Yeah, hello. So please feel free to interrupt me anytime and ask questions. I will tell you about our technology, I guess, computational technology to model DNA nanostructures. So in general, my lab works on all kind of nano devices. In addition to biomolecular systems, we work a lot on nanopores and on DNA nanostructure. So I guess I decided to make it more focused. So I'll tell you about our DNA nanotechnology work and to introduce three tutorials. So you don't have to do all three. Hello. Good afternoon. Yeah, so there will be three of those tutorials and two senior person from my lab will be available to get you started. I guess it's probably not feasible to do them in an hour and a half, but at least, well, actually one of them could be done. Okay, so let me see how I hide this thing here. Here it is. Okay, so let me start with introducing what structured DNA nanotechnology is. You might have heard of it. It's so-called DNA origami. So in a typical experiment, you would take a long strand of a viral genome. So this white thing is a single-strand DNA. And then let's say you want to make a sculpture out of it. Of course, the sculpture in this case will be the logo of the University of Illinois. So the way you're going to do it is by adding so-called staple strands that will bring fragments of the DNA that are distant along the genome into contact, so through hybridization. So you'll take a staple strand. It's not drawn to scale, so one part of it. By the way, can you see my cursor?
Yes.
Okay, great. So one part of this strand will hybridize to one part of a structure, the other to the other. And then you'll add lots of those strands like that. And an experiment, you raise the temperature, you mix it up, and then you gradually cool it. And as you cool it, it will assemble into the structure that it was designed to assemble, in this case, the logo of the university. So this is something that's been around since 2006. So following pioneering work by Ned Seaman, so Paul Rosamond invented DNA origami, and then it has been applied to all kind of systems. So it works extremely well. So if you want to build a system from DNA, you can. You just have to design it. So here's some examples, 3 images of designs, and those are the actual images obtained using electron transmission microscope. It works extremely well. So computer models were at the very beginning of the field. So when Paul Rosamond designed this origami, 2D origami structure, so one part of the process was writing a code, that would select those staple strands. So when you add them to a viral strand, it will fold them in the design structure. So the field really greatly benefited from this CAD Nano, which is a kind of a graphics design program that allows you to build a 3-dimensional DNA origami structure. So that code was written by Sean Douglas, who is now at UCSF. And that's pretty much a big fraction of the field uses that software, CAD Nano. There are, of course, other ways to build DNA nanostructures. One is that nano engineers like click and connect kind of tool. So you can build things in it. vHelix is actually a plugin for Maya. It's kind of an advanced DNA design tool. Tiamat works under Windows, and there's a suite of programs from Mark Bath lab at MIT, mostly targeting development of this wireframe structures. So we are interested in modeling DNA structures, and before I describe what we do, I have to put it in perspective of what people have done. So there's really a spectrum of approaches one can use. So on the left here, you see like a finite element, like mechanics type of model of DNA called CANDO from Mark Bath lab. Then you have coarse grain models like OxDNA, which is a really good coarse grain model of DNA. Those two are our models, EnergyMD and AtomMD, and of course each of them has a target range of applications and comes with a price in terms of computing. You know, with continuum models, you can run them through a web interface, and they're relatively cheap to do. Coarse grain models require GPU workstations, and they are somewhat involved, but you can have a run done overnight. And then finally, we have our all-atom approach, which requires a high-end computer machine. Okay, so what are the typical problems in the field, and what do we modelers offer as a solution? You know, one of them is, I guess the question is like, what is it, right? So you have a design of a nanostructure, and design is what you see here. It's like, you know, those rail tracks. That's how a CAD nano design looks like. Of course, when you design CAD nano, it also has a graphical interface, so you can kind of see what's idealized structure is in 3D. But that is an idealized structure. It's not the actual structure in solution. But with our multi-resolution simulations, we can predict what the structure of the object is in solution, and with the accuracy that approaches or exceeds that of cryo-EM. So another type of problems is you have an experimental collaborator who has come up with a creative way of using DNA origami. Like in this case, it's a voltage sensor, but it's not really clear like why does it work or how does it work. Well, then you can just go and build a model and extract experimental observables from your simulations and relate it to microscopic processes that actually happen in the device. And then the final, I guess, the most interesting and rewarding part, but also one of the most difficult one, is where you use the computational approaches as a way to design new systems of capabilities that have not been explored experimentally yet, which is this case of a DNA scramblase, where we first computationally predicted and then experimentally verified the ability of DNA nanostructures to scramble lipids. So typically DNA origami process looks like that. You have an idea first, and then you go into this CAD nano and you build a structure. You get a schematic model from the CAD nano itself. Again, it's like idealized, so you have straight helices, right angles, and so on. The software predicts the library of sequences that you need to buy. And in principle, this is where you can go and do DNA synthesis. Of course, nobody does DNA synthesis themselves. You just order them. So you get a bunch of samples. You run the assembly, then you verify it by doing gel electrophoresis. Typically, that is a good indication of whether or not the structure folds. And then you characterize it. And with characterization, well, there are a few things you can do. You can take AFM images, or you can do TM images, and sometimes you can do cryo microscopy reconstruction. So the typical cycle is about 3-4 weeks. But with this multi-resolution approach, we can do it in less than 24 hours. So there are several ways to characterize experimentally nanostructures. So gel electrophoresis, I already mentioned. So the bands here are the DNA constructs, and you can kind of tell how far the DNA construct travel in electric field by how far they are from this DNA ladder.
think anything out of the sheet is it, but I can do it now.
And you can use atomic force microscopy, transmission microscopy, fluorescence. But the only really way to get 3D structure is through cryo-EM. So somehow those DNA origami structures, they do not like to crystallize. Actually, they're floppy enough to diffract sufficiently enough to build the cryo-EM reconstruction, to build the 3D reconstruction through diffraction patterns. Cryo-EM is the only way to do it. And of course, there is a computational microscope. I guess it was introduced many times. You already know a lot of molecular dynamics simulation. So here's my take on it. So it's kind of a way to get movies from microscopic systems. So here's one of the last one I made myself. You see a DNA molecule going through the nanopores. That's for our nanopore sequencing work. So we were interested in seeing how well our computational microscope performs in studying those systems. And when you basically enter the field, and that's what we did back in 2012, well, the first thing was how do we build those systems, right? Because design is like that. It's like flat thing, schematic, something like that. So we wrote, I mean, Jijung, a former postdoc, now a faculty in Korea, so he wrote this fairly popular tool called CAD Nano to PDB. So what it does, it takes this design and it builds an ideal all-atom structure. So for many people, that's pretty much what they want, because they want to make a picture out of it and have a picture in a paper. But we can do more. So we can put it in water, we can add ions, including magnesium ions, and we can simulate. So this should start the movie, maybe. Yeah, it doesn't. Okay. It's gonna work. Okay, now it's working. So you see, so we can simulate that using all-atom molecular dynamics method, and we can observe how the structure deviates from its original ideal shape. You can also clearly see those like chicken wire patterns. So you have like helices together, helices apart, and that is something that was seen in an experiment. More, we can take those simulations and analyze them in the framework of Frenet analysis. And this is something, if you're interested, you can read in this paper. But basically, the framework looks at the local fluctuations of the DNA origami nanostructures in order to extract local mechanical properties. So we, for example, can compute persistent lengths of those objects and see how they vary depending on the design. So we have one tutorial, and that's called a practical guide to DNA origami simulations using NAMD. So if you already know how to model things using NAMD, you can try to do that. It starts with a CAD Nano design, and then you end up with this basically all-atom structure. You can do it without installing CAD Nano, but I think you'll get more out of it if you, like, if, for example, you want to do it for your own design, if you get CAD Nano is available for free. So, of course, the field is not limited to origami. So here's another example where instead of having one very long virus fragment, which is origami, you can have like lots of many short fragments, so-called DNA bricks. So those can also be self-assembled into 3D structures of arbitrary shapes. And we also have had a service that enables modeling those DNA bricks at NanoHub. All right, so does the method really work? So that's kind of a question that you need to always ask when you go to molecular simulations. So one way to do it is to directly compare results of cryo microscopy with petascale computer reconstructions. So here's the design, and here are the experimental features of the design. So in experiment, you average lots of those fragments to get a consensus 3D structure to arrive at a pseudo-optimistic model. So this one is not molecular dynamics, it's the one that you can update by fitting things into cram density. You can do it using MDFF, for example. But you can also just build your atom structure and simulate it in water. So this is a fairly big system, 7 million atoms. Was one of the biggest systems simulated back then. But what do? We ran it, and look, magically it starts crooking in the right direction. So this is all atom simulations with all ions and waters and everything. So within about 200 nanoseconds, you get something that looks reasonable. You can look at the same system from a different side, and yet another one. So at the end, we can take our structures and make a density from it, and then ask the question to you, which one of the two is the experimental one and which one is MD derived. And of course, you probably can tell. Okay, the left one is a cryo-EM, the other one is MD. They are really, really similar. So if you kind of look at the comparison of the two, you'll see they're right on. So basically, the blue one here is the MD derived structure. It's slightly longer because it has those single-stranded overhangs that are not resolvable in the cryo-EM reconstruction because they're just too floppy. So this the pattern of crossovers is kind of reproduced extremely well in MD simulation. Also, the overall kind of so this is part was designed on purpose to see how those densely stacked junctions look like. And also look at the overall geometry, and you see it has this global warp. And at the end, the DNA helices, if it's missing a crossover, they like to kind of split up. And overall, it actually works really well. The only problem with it, it's a hero simulation. So if you don't have a petascale computer or like a larger sub-petascale machine, well, you are out of luck because you cannot do it. So that kind of bothered us a little bit. So we decided to see if we can simplify the system now that we know the result. So what we did is kind of you can call it a implicit model type of approach. So we take our all atom simulation and we run it in vacuum. So it's all atom in vacuum. But what we do, since we know the local distance, like what should be the local distance between helices, for example, if you look at this image, like there's a crossover and then there's this chicken wire pattern. So we can enforce it using harmonic restraints. And if we do it, it's actually shockingly how well it works. A, we don't have to simulate water, so it's really fast. And B, it relaxes like 100 times faster. So overall, we get maybe a factor 10,000, maybe 100,000 speed up. Look, in 1 nanosecond, it's right on already. So this method we called elastic network restrained molecular dynamics. I forgot what it stands for, but energy MD sounds really cool, and it's been used by experimentalists to test various design at multiple stages of the process. So if you don't have access to petascale computer, but you want to simulate those larger systems, you can do it using this method. We even have a web server that we don't run things for you. We just make everything for you to run it. Just go there, click, get an archive, but you have to run it on your own resources. So it was of course nice, but some of the structures have those programmable bands. For example, here you're looking at an L structure, which has missing parts in the DNA. So it enforces us to bend by 90 degrees. So back in 2013, we could do it. in a huge molecular dynamic simulations. But again, this is something that only a few labs in the world can do. Unfortunately, our energy MD is not really good for this kind of simulations because it's still very slow because you simulate every atom. But in the field of origami design, many of the structures are hollow. For example, look at this kind of buckyball, it has mesh, it's mostly water. So also in principle, you can run a simulation of like that using all atom MD, but it's going to be kind of a waste because you end up modeling mostly water for a very long time. So we had to think of something else, and that's how we come up with this multi-resolution DNA method, Mr. DNA, and Chris Maffio, who is Mr. DNA himself, will be available in the afternoon to tell you how it works. So, by the way, this multi-resolution DNA model is not limited to origami. We have successfully used it to model packaging of viral genomes, nanopore transport, all kind of things where you have origami, you can, and where you have DNA, you can definitely use it. So the idea here is that we will trade resolution for computational efficiency, starting with something very coarse, and then as we progress, we'll refine the details. So this is how it works. So at the core of the model is this multi-resolution model of the DNA itself. So now here you're looking at the same fragment of DNA modeled at different resolutions. So what's essential here is that the physics is the same. So you get the same persistent lengths, same non-bonded interactions, everything the same, just like in all atom resolution. The only difference is that you vary the level of details that you describe your DNA fragment. So it's kind of essential. Our non-bounded interactions are calibrated against all atom simulations and experiment, and regardless of the resolution of the model, we get the same forces between DNA. So it is a pretty good model. We also do not allow strands to cross, which sometimes you find in other coarse grain models. So this is how it typically works. You start with a nano design. Again, it doesn't have to be nano design. You can build anything you want in mRDNA or any other builder program. Then we relax it at low resolution. Then we gradually refine it, introducing twist, going to 2 beats per base pair, and eventually ending up with the all atom structure. And this all atom structure, you can actually take and add solvent and run it. It's an MD ready structure. It doesn't have clashes, and you can either run it with a all atom MD with solvent, or you can do energy MD on it. It runs just out of the box. Let me show you some examples. So here's again this pointer structure that we have the cram structure for. So we start with a low resolution, then we kind of switch to a finer, coarser model, and this is energy MD simulation. It is really fast, has the same accuracy as prior reconstruction, and also another coarse grain model is OxDNA. It's just a lot faster because it has fewer beats, and the relaxation is streamlined in terms of resolution switching. So we have used it to predict results of cryo reconstructions. It's right on. So the reason we developed is to see if we can, to find an efficient way to model curved structures, and of course we can model curved structures with it. We can model structures that are kind of difficult to model using anything else. So here's like a box. It's kind of a jewel box that can be opened and closed, but computationally it's difficult to model because it's a hollow structure. So through the steps of simulations, we get the all atom model. We actually use the all atom model to improve the design of those boxes and interpret fret measurements that's described in this paper. So here's another difficult design. It's called Flask. So once you take the Cardano design and you get the coordinates basically flat, which is not good because it will collapse, but we have a way to unwrap the coordinates, and then you would get like a really representative confirmation of the DNA flask. Again, we start with a low resolution model, going through all of the steps until you end up with a fully atomistic structure. So the mRDNA package, it actually has you can modify it, but it gives you a number of default choices of how to run it. You can tweak it a little bit, but I think the ones that we have there by default work for almost for all cases, just out of the box. It's not limited to DNA origami design. So there's this wireframe design. This is a bunny from Bjorn Hogberg lab and at Karolinsky was it's a cover of Nature at some point. Here you just give it a tessellated design. It will automatically determine the sequence of strands that you need to mix to get a 3D structure. In this case, it's a bunny. So you can read natively the helix design or any other design that is there and simulate it. Nanomechanical systems. So here's some kind of a gear where you have a connection between motion along the shaft and this kind of a thing here. This is the Bennett junction. Again, here you have a topography reconstruction. Here you have a simulation. It works pretty well. It's not 100% like quantitatively accurate because our model of single-strand DNA is not ideal, but it's pretty good. So at the hood of the mRDNA package is ARBD, which is our atomic resolution Brownian dynamics simulation software, which is developed by our NIH center. So it runs extremely well on GPU, very efficiently. We've used it to model transport within the organelle, the photosynthetic organelle that we published in Cell 2 years ago. It's a fairly general package. You can model chromatin, nuclear pore complex, and many other things. I guess the good and the bad thing about it runs on GPU. So it runs very fast, but you need a GPU to run it. So if you want to do this multi-resolution simulations, you have to have either a laptop or a workstation with NVIDIA GPU. Otherwise, it's not going to work. So the software is available on GitHub, and also you can find it on my lab's website. So my lab's address is over here. I will have a separate slide for it, so you don't have to rush to write it down. So we have a tutorial, multi-resolution simulations of self-assembled DNA nanostructures. You can find it on GitHub, or you can find it at my lab's website, which is bionano.physics.illinois.edu. So let me show you one application of the mRDNA package to real science problem. So sometime back so experimentally it's possible to use DNA origami nanostructures for ionic current measurements. So in typical experiments, you would have a pipette. You see there's a pipette, and there are two electrolyte solutions. Everything is in a PDMS matrix. You have electric field applied using these electrodes and the voltage, and you can also measure the current that flows through it. So you put those plates in one compartment, they will get captured eventually, like this. And once they're captured, the current that flows through the pipette would be reduced to a certain level. So we were able to model those processes using all-atom MD, and that's something that you learn how to do from our first tutorial. But that's not the point. What we discovered here was that when you apply electric field, we also deform the origami. You see, it's kind of becomes like a nano accordion. So the idea was, well, maybe we can use this deformation as a way to measure local voltage. So we can design sensors of voltage and put them on neurons and maybe measure action potential as it happens at the single molecule level. So this is how we used our mRDNA package. So one thing that it has that many other packages don't, it has this ability to couple to continuum models. For example, COMSOL. If you ever used COMSOL, you can just take geometries that you build there or external electrostatic potential and just input it into mRDNA. So here's the solution from a COMSOL, and we can import it in the simulation, creating a model of a pipette. And then we have already our model of the origami plate, and we can apply voltage and see what happens, how does the plate deforms in those voltages. By the way, if you're interested how we made those movies, we also have a tutorials on that. Some people like to know how it's done. And so I'm not showing you this guy at 600 millivolts. It's actually a funny one. Look what happens. It actually slides through. The forces from electric field are so huge that it deforms it. And that is exactly what you see in an experiment. If you do experiment at 600 millivolts, all of them just pass through the entrance. So once we have this trajectory, we can look at the fluctuation between the dyes that in experiment report on the local deformation of the field, and we can do interpretations of the experiment. So this is not the point here. The point here is that you can run simulations at different conditions that are accessible to experiment, and you can analyze the result, drawing conclusion about experimental outcomes. So in this particular case, we showed that this volt set is possible to read out local voltage on the origami plate by measuring Fred signals from the dyes. All right, let me slowly go to the last subject of my talk, so-called the DNA membrane channels. I guess some of you are interested in modeling membrane channels in general. So, we have this membrane channel tutorials that we've written some time ago that is now that Emmett's lab now maintains. But those are classical membrane channels. So it turned out that one can build membrane channels out of DNA. So here you see a zoo of membrane channels, and many of them we've built and designed in collaboration with Ulrich Kaiser from Cambridge University in UK. So here's a typical biological membrane. Yeah, you have this very hydrophobic part inside and hydrophilic head groups. So when I said DNA ion channel, you might think I'm crazy because, of course, you know that DNA is a hydrophilic molecule. So it doesn't want to go into a lipid membrane. And that is true, it doesn't. But you can make it go. Basically, the way to do it, you decorate the nanostructure with cholesterol anchors. So you see those red things here, those are cholesterol that are attached. And if you have enough of them, it will be sufficient to compensate for the energy penalty of having this negatively charged thing inside the bilayer. So there's another way to do the same. You can take the DNA strands and you can modify its backbone. So backbone is usually negatively charged. So once you chemically modify it, you basically can neutralize them, or you can neutralize them and make hydrophobic. So there's a whole range of possibilities of how to do it, but that's another way of doing it. Yet another one, you can add this bulky porphyrins, which are another hydrophobic moieties to the DNA bases themselves. So all of those have been realized in experiment. So we were kind of really interested in those type of systems because we're expert in membrane channel simulations. We wanted to see what would it take to build, for example, a DNA syringe, a syringe that will inject maybe some kind of medicine into cells. So here it is. It's a big DNA origami structure made of 112 helices that are decorated with cholesterols at the bottom over here. So in order to simulate this, we kind of go through all the steps of procedures where we start with a nano design, we relax it using energy MGs, and we put water ions, lipid bilayer, cholesterol anchors, and we can apply electric field and measure ionic conductivity. So once we measure ionic conductivity, we can compare it to experiment. It's actually right on in this case. Yeah, in this case, the experimental conductivity is 13 nanosiemens. And that's exactly what we get from our simulations. So we were interested in building the smallest possible, simplest possible DNA channel. The DNA channel that actually does not have a channel. So look, here it is. We just took one single DNA duplex and decorated it with porphyrins. So it looks like that, surrounded by water ions. And once you run the simulations, you'll see something interesting. You have this kind of a toroidal pore that is forming around the DNA structure, right? While those anchors penetrates through the head groups and not let the DNA nanostructure leave. So think about it. We just made an ion channel from a DNA helix that doesn't have a hole, because the hole is around the DNA. So in this particular case, we can match experimental conductance quantitatively. Simulation experiment, pretty much about the same. So there was one other thing that came out from the simulation. We observe, and that's something that you cannot do right away in experiment, that something funny happens to lipids. Look, here's one guy who was at the beginning of the simulations in the upper leaflet. It will then spontaneously move to the lower leaflet. And the reason why it can do it, because now, once we place the DNA into the membrane, we kind of connected the top leaflet of the bilayer to the bottom left leaflet of the bilayer. So now the molecules can diffuse both ways and scramble lipid composition. So this turned out to be kind of a big deal, because if you look at any biological membranes, they all have asymmetric composition. And biologically, it makes sense, because if your cell, what's inside is different than what's outside, and you want to decorate your membrane with different lipids. In biology, there are special enzymes that scramble lipid composition called scramblases, and that usually happens when something, like when you want to incorporate vesicles, for example. It's also a process that leads to apoptosis, meaning the programmable cell death. So the scramblases, they scramble lipids, but they do it very inefficiently. Our DNA scramblase turns out to work about 10,000 times faster than any protein enzyme known today. So bad things happen if it doesn't work. So one of the application could be to create a therapeutics based on that. So just a few words about how this kind of things can be measured in experiment if you're interested. So experimentally, the classical assay is to add a lipid that has a dye. And that dye can be quenched, meaning you will not see fluorescence if the small molecule, dithionite, can reach it. So the idea is that dithionite by itself doesn't go through the membrane, because it's highly negatively charged. So the idea is like that. So you create a lipid vesicle. So you have those labeled lipids on both sides, both leaflets of the membrane. Then you add dithionite, and then you should end up with an intensity that is about half of the initial one, because dithionite cannot go through the membrane, but it can still render those fluorophores inactive that are exposed to it. Now, in terms of if you have scrambling, so the scramblase will kind of scramble lipids. So this way, each of the lipids will have a chance to be exposed to that ionite, and intensity will go to 0. And then you can also have a non-scrambling control where its DNA kind of attaches to the bilayer, but it doesn't scramble because it doesn't penetrate the bilayer. In this case, you expect the intensity to go to 0.5. So, and this is exactly what we see in experiment. I'm gonna go quickly through it. So when we have a DNA scramblase, we see the intensity trends is going to basically 0, indicating scrambling, and for non-scrambling control, it goes to about half. So there's one of the kind of you can ask, well, so what? Like, is it used for anything? So one possible application of this method is actually in killing things. So, in normal healthy animal cells, those PS lipids are usually on the inside of the membrane. And you know it because there's this protein called annexin 5, which binds specifically to PS lipids. So if you have healthy cells, basically you don't see any signal because there's nothing for it. There's no PS lipids for it to bind. Now, if you chemically induce apoptosis in the cell, you'll have those PS lipids exposed, and you will see it as a light up here because the annexin 5 will bind to it. So what we found was experimentally that if we now expose a cell to a scramblase, the scramblase will scramble PS lipids to be outside that you can see them with a stain. And usually having PS lipids outside is a signal saying, okay, I'm a cell, I have a problem. I guess, can someone please terminate me, So this is a way, completely orthogonal way to work with cell homeostasis that wasn't on a horizon before, because, well, there was no way to introduce scrambling on demand. And with those DNA nanostructures, we should be able to target the cells that we want, because of course we don't want to produce scrambling in healthy tissues. And one other thing that we need to do before we can use it for therapeutics, we have to make those scrambling switchable, ideally photo switchable, so you can not only target them to the cell or to the cell type that you are interested in targeting to, but also you want to localize switching by, say, UV light or something like that. So if you are interested in modeling DNA nanopores, we have a tutorial for you. So this one is similar to the first one that I showed. It's an all-atom molecular dynamics tutorial. The difference is that the system that we describe there is a 4-helix bundle. It looks like that, but it also has those cholesterol anchors with a linker. So this tutorial describes how you go from this idealized design to having DNA origami, or I guess no, it's not origami, self-assembled DNA nanostructure embedded in the lipid bilayer membrane. Steps required to calibrate it, and then you can apply electric field and see ions actually going through and compute the current voltage curve, something that you can directly compare to experiment. Of course, if you run it long enough, you also see lipid scrambling, which is kind of cool, but that requires a microsecond long simulations, which now I guess you can do with an MD3 and GPU. That won't take too long. Okay, so so here's what we have for you. We have 3 tutorials that I kind of covered in this talk. So in talk, mostly focused on why we're doing it, how we're doing it, and in very broad strokes, and yeah, and what are the kind of typical scientific results that comes out of it. Now, the tutorials, they are really kind of step by step showing, okay, this is how we do it. We start with step 1, step 2, step 3. So the multi-resolution simulation of self-assembled DNA nanostructure. So that was the second part of my talk, where we show how we start with the idealized design and end up with the all-atom structure. So the tutorial is actually very flexible. You don't have to have a DNA nanostructure. You can just have a long piece of DNA, or you can also read in a PDB. So if you have a PDB of DNA that you want to simulate using our multi-resolution approach, it should work. You should just be able to read in your PDB and run it. So the only issue with this tutorial is that you need to have a GPU card. If you have an NVIDIA GPU card, it's going to work. So Chris actually wrote both the tutorial and the package, so he'll be available to help you with this if you're interested. So then we also have a practical guide to DNA origami simulations using NAMD. So that one describes how we do all atom simulations. It has a part on energy MD. It's like one of the steps before we do the full all atom simulations that could be useful if you are interested in modeling those kind of systems. So both Chris and Himanshu are expert in origami simulation. They will be able to help you with that. And then the final one is this all atom simulation. So membrane spanning DNA nanopores. That's the last part of my talk. So we have an example of exactly this 4-helix bundle, same one that we use for our scrambling paper. You can see how it's done. This example, actually those both examples are good, provide good illustrations to our custom model of magnesium. You know, ions are very, very important in simulations of DNA, and we have reparametrized ion DNA phosphate interactions, and we have a separate actually page on it. But if you actually want to know how to use it in practice, in both of those tutorials, we use our most up-to-date model of okay, I am going to finish here acknowledging various funding agencies and people who have contributed to the talks that I've just shown. So I already talked about those 3 gentlemen here. So Chen Yu Li was a biophysics student in my lab who graduated some time ago. So he did the scrambling simulations and the pioneering work on DNA origami nanopores. Okay, I'm done, and I've been talking for now like 50 minutes. I guess, I hope there's still, I didn't like lost connection or anything.